Assalamu alaikum. I am Dr. Naurin Khan, lecturer of Biochemistry department. Today I will discuss about enzyme. Enzymes are the protein biocatalyst produced by the living cells and which will increase the rate of chemical reaction and are not consumed during the reaction they catalyze. Normally, enzyme cannot initiate the reaction. They will accelerate the rate of reaction, and at the end of the reaction it will remain unchanged. What are the properties of enzyme? Properties of enzyme. Enzymes are the protein in nature except ribozyme, which is RNA in nature. Enzymes are water soluble, non-dialyzable, colloidal, and they are heat labile. Enzymes have definite isoelectric pH, and enzymes are the biocatalyst. Now come to the point, enzyme specificity. What is enzyme specificity? Enzyme activity, which is specific for substrate and specific for definite type of reaction, that is known as the enzyme specificity. There are two types of enzyme specificity: substrate specificity and reaction specificity, because enzyme specificity depends on the substrate and the reaction. So what is substrate specificity? Substrate specificity is when the enzyme activity depends on the substrate, and when the enzyme is specific for a specific type of substrate, that is the enzyme specificity. There are two types of substrate specificity. Number one is absolute specificity and number two is reactive specificity. Absolute specificity means definitive enzyme specific for definitive substrate, such as glucokinase enzyme. This is specific for glucose. Urease enzyme, this is specific for urea. So this is the absolute enzyme specificity. And now come to the point, relative specificity. When there is a one enzyme, but due to the structural similarity, it can act on all structural similar kind of substrate, that enzyme is known as the reactive specificity. Such as trypsin is an enzyme which act on different type of protein. So this is the reactive specificity. Now come to the point, reaction specificity. Reaction specificity means such as from one substrate it can produce different type of product, but in case of different type of reaction the enzyme will be different. Such as from pyruvate to acetaldehyde, when formed, there will be the pyruvate decarboxylase enzyme. But when from pyruvate there will be production of oxaloacetate, the enzyme will be changed. That is pyruvate carboxylase enzyme. So this is all about the enzyme specificity. Now come to the point, what is coenzyme, what is cofactor? Coenzyme. Coenzymes are organic substances, low molecular weight, heat stable and non-protein in nature, and it helps in the enzyme activity. When coenzyme tightly bound with the enzyme, that is known as the apoenzyme, and coenzyme and apoenzyme combinedly complex form, that is known as the holoenzyme. There, most of the coenzymes comes from the vitamin B complex derivatives, and some are non-vitamin B complex derivatives. From vitamin B complex derivatives, coenzymes are from thiamine, vitamin B1, from thiamine the coenzyme comes, that is thiamine pyrophosphate. From vitamin B2, riboflavin, the coenzyme comes FAD-FMN. From niacin, the coenzyme that will be NAD and NADP. From pantothenic acid, the coenzyme is coenzyme A. From pyridoxine, coenzyme is pyridoxal phosphate. From folic acid, tetrahydrofolate, and from cobalamin, methylcobalamin or 5'-deoxyadenosine cobalamin. These are all the coenzyme which comes from the vitamin B complex derivatives. And there are some coenzymes which are not vitamin B complex derivatives. They are coenzyme Q, lipoic acid, etc. Now come to the point, cofactor. Cofactor are inorganic metallic ions which helps in the enzyme activity. For carbonic anhydrase enzyme, there is a cofactor metallic ion, that is zinc, and from catalase, peroxidase, cytochrome oxidase, for these enzymes the cofactor is iron. Now come to the point, what is the active site and allosteric site for enzyme activity? When the substrate bind with the site of enzyme, that is the active site of enzyme, and other than the active site where the allosteric inhibitors will bind, that is the allosteric site of enzyme. So in an enzyme there are two sites, active site and allosteric site. Now we will discuss about the Iub classification of enzyme. Iub means International Union of Biochemists, which started in 1964, and they classes the enzyme according to the type and mechanism of action of chemical reaction. Now come to the point, what are the classes of enzyme? First classification is oxidoreductases. This group of enzyme catalyzes oxidation and reduction of the substrate, such as from lactate through the lactate dehydrogenase enzyme there will be the production of pyruvate, and here are the enzyme dehydrogenase, catalase, peroxidase, oxidase. These are all the oxidoreductases group of enzymes which catalyzes the oxidation and reduction. Now come to the point transferases. Transferases, this group of enzyme catalyzes the transfer of functional group other than the hydrogen group from one substrate to the another substrate, such as alanine aminotransferase, aspartate aminotransferase. And there is the example from serine, there will be the glycine. There is the transfer of the functional group, so these are the transferase group of enzyme. Now come to the point hydrolases group of enzyme. This group of enzyme catalyzes hydrolysis when the bond will be cleavage with the presence of water and there is no coenzyme is needed, that is the hydrolases group of enzyme. And most of the digestive enzyme in our body are the hydrolases group of enzyme, such as lactase will hydrolyze lactate from lactate to glucose and galactose with the help of lactase enzyme. And when fat will be break down by the process of hydrolysis from fat we will get fatty acid and glycerol with the enzyme lipase. So these lipase, lactase, sucrase, amylase all are the hydrolases group of digestive enzymes. Now come to the point liases or synthase. This group of enzyme catalyzes splitting of the large substrate, leaving the small group by non-hydrolytic cleavage and leaving the double bond. Here linking of the two molecules without expenditure of the ATP. So this group of enzyme are known as the liases group of enzyme. There is a example from pyruvate, we will get acetaldehyde with the help of pyruvate decarboxylase. Here the decarboxylase is the liases group of enzyme. Besides the decarboxylase, there is citrate synthase. These are also the liase group of enzyme. Now come to the point isomerases. This group of enzyme catalyzes the isomerization and formation of the isomer of a particular substrate. And the enzymes are isomerases, epimerases, mutase, such as from methyl malonyl CoA there will be production of succinyl CoA by isomerization and the enzyme is mutase. Now come to the point ligases. Ligases is a group of this group of enzyme will catalyzes the linking of the two molecules with the expenditure of ATP. In the liases where there was no expenditure of ATP, but in the ligases group of enzyme there will be the expenditure of ATP, such as from pyruvate when there will be the production of the oxaloacetate, pyruvate carboxylase enzyme is needed. This pyruvate carboxylase enzyme is the ligases group of enzyme. So ligases group of enzymes are DNA ligase, carboxylase, etc. So these are all about the Iub classification of enzyme, and there are main 6 classification. Number 1 is oxidoreductases, transferases, hydrolases, liases, isomerases and ligases. These are all 6 classification of the enzymes. So today's discussion about enzyme up to here. In the next session we will discuss another topic from enzyme. Thank you all for your patience and hearing. Allah Hafiz.